There's a global shortage of donor organs, particularly hearts. Many patients are dying while waiting for a transplant. One of the proposed solutions is xenotransplantation, which involves transplanting organs from an animal into humans. Pigs are considered an ideal donor because their organs are similar size and function to human organs, and importantly, their genomes can be genetically modified. In this presentation, I'll explain the molecular basis of the immune rejection, how pig hearts are genetically engineered to overcome this, and what recent human cases reveal about the limitations of this technology.
The main barrier to xenotransplantation is the human immune response. Pig cells contain a carbohydrate molecule called an alpha-gal, which is normally produced in most mammals by an enzyme that humans don't have. Because humans lack this enzyme, our cells never express alpha-gal, so the immune system recognizes it as foreign and already has antibodies against it. When a pig heart is transplanted, these antibodies bind to the surface of the organ, especially the blood vessels. This activates the complement system, which damages the cell and triggers the inflammation and clotting. As a result, the blood flow is disrupted and the organ rapidly loses function. This leads to a hyperacute rejection, where the heart can fail within minutes and sometimes causing the heart to turn black and die within seconds. To overcome immune rejection, donor pigs are genetically engineered using tools like CRISPR. CRISPR cuts specific sections of DNA so genes can be removed or changed. One key change is removing the gene that produces alpha-gal. Without this gene, pig cells no longer display this molecule, which reduces immediate recognition by the human immune system. Other genes that produce similar molecules are also removed to further reduce the antibody binding. Human genes are then added into the pig's DNA. These genes produce proteins that can help the immune response, reduce inflammation, and prevent abnormal blood clotting in the transplanted heart. Another key change is removing the growth hormone and the receptor gene. Without this receptor, the pig's heart does not continue to grow after the transplantation, which helps maintain the normal heart function. Overall, these changes make the pig heart more compatible with the human body. The clinical potential of this technology has been demonstrated in two recent human cases. The first involved David Bennett Sr., who received a gene-edited pig heart in 2022. He survived the critical early period where the hyperaccurate rejection would be normally occurring, indicating that the genetic modifications were effective at preventing immediate immune destruction. However, his condition worsened over time, and he ended up dying 2 months later. A pig virus known as porcine cytomegalovirus was later detected, which may have contributed to the graft failure. The second patient, Lawrence Fawcett, received a similar transplant in 2023. Initially, the heart functioned well, but around 2 weeks after surgery, rejection began to occur. This was due to the antibody-mediated rejection, where the immune system generates new antibodies against the pig organ, leading to endothelial damage and organ failure. These cases show that while some hyperacute rejection can be controlled, long-term immune responses remain a major challenge. Despite significant progress, several limitations remain. One major issue is the risk of cross-species infection, particularly from porcine virus. Even with genetic engineering, the adaptive immune system can still generate new antibodies over time, leading to a delayed rejection. These also raise ethical concerns regarding the use of genetically engineered animals for organ harvest, as well as questions about cost and accessibility. While these challenges are not unique in xenotransplantation, they highlight the complexity of translating this technology into routine clinical use. In conclusion, genetically engineered pig hearts represent a solution to the global shortage of organ donors. By modifying key genes, scientists can prevent immediate immune rejection and allow these organs to function temporarily in humans. However, long-term success will depend on overcoming immune, infections, and ethical challenges. meaning this technology is not yet a complete solution.